শুরু করছি।
Okay, from the last class, we see about the comparative representation of gel electrophoresis and capillary electrophoresis. Any question here? If no, for example, in our case means in DNA fingerprinting case, you have to run multiplex PCR together. You have to run simultaneously multiple samples or multiple locus PCR together, and that will present something like this type of messy structure, messy representation. For example, here you have many peaks and you don't know which one is which. So this could be identified like you may have like several different PCR results here together. And when the PCR is done, the machine will represent you something like this. But in your like notebook or somewhere, you have the data like I am doing the PCR and this color should represent this specific number of locus that I have checked. For example, here you can command the software to keep all the red peaks and remove all the just a single command so that you can understand. For example, like if you have two different colors for many different type of PCR, like here you have blue and red together. So you just command the computer or the software that you just want to see the red color peak. The software will remove or obscure all the blue color like this. So now you have only the red color results. And from this, you can understand which one is red color result and what is the result. For example, here you can see 1, 2, 3, 4, and 5 peaks. And in your case, or in case of DNA fingerprinting, you know that you have, like for heterozygous, you should have 2 band or 2 peaks. And if the sample or locus, you should have single peak. For example, here, this one probably one result like one locus result, which represent a single peak. This and also another locus result, which represent a single peak. This very two nearby result or nearby peak, it should be a heterozygous locus result. দুইটা peak একসাথে কাছাকাছি, মনে করো 15 and 14 অথবা 15 and 10 repeats together. So for 15 you have one peak or one band. In case of normal electrophoresis, it is band. In this case, it is peak. For 15, you have one band or one peak, and for 10, you have another band and another peak. So this result represents it is kind of like heterozygous result, and this one also shows a single peak. It is also represent a single peak. That's why it is maybe a homozygous result. By looking at the size of the peak, for example, from 0 to 150. So it is between 150 to 300. Just you can say this is like 180 or something. This one exactly 320 or something. This one between 470, 480 or something like. Okay, let me have this. যা বলছিলাম যে তোমার এখানে পিকগুলা দেখে you can understand about the size of the product and also the intensity of the product. So this way you can command the software to keep, like in this case, all the red color product here, or you can command the software like keep all the blue color product, something like it will then omit all the red color product or all the red color pick from here. So by this you can get an idea about which are the green color, or in this case blue color product in your complex result. So by this way you can get each and every result one by one by one by one. So it is a very easy way. When you are using the software, you just have to get the command, like you can separate them according to the color, or also you can separate them according to the like size. For example, if you command the computer that just keep all the band which is less than 300, so it will then omit all this more size band. For example, these are more than 300, it will remove all the this sorry all this bigger size or longer size band. Like you command keep all the 300 or less than 300 size band and omit all the more longer size band, it or peak, it will remove the longer size peaks from your. So from here you can get all the peaks that is presented less than 300 in this diagram or in this graph. By this way, if you know about your result, what should be your result? For example, you have a locus, it should have a size like between 100 to 150. Then you can command the software that keep all the band between 100 and 150. Then it will show you just one or two or whatever the peaks are there. From that you can get the result. Or you can command the computer or the software that keep only the red color band, only the blue color band, or only the yellow color band or peaks. Then it will do according to your desire. By this way, from a message structure means like it will show something like this type of structure, many peaks. For example, this is one of the peak, so many peaks together. From here you can command and keep only the thing that you need. By this you can get the real result of your each and every locus separately, one by one by one. So this is the way that people are doing the capillary electrophoresis. And as you see that the normal DNA, the DNA that we are handling, is not useful. Normal DNA means all the DNA which is not labeled, by the fluorescent dye is not useful in case of capillary electrophoresis. So you have to have a method to label the DNA. And to label the DNA, we can do the fluorescence tagging, and there are three different methods of tagging or labeling the DNA, methods for labeling the DNA, and there are three different methods. I will discuss these three methods one by one. Before going to the method, if you have any question about this type of like gel separation in capillary electrophoresis, you can ask. Okay, if there is no question, we can move to the labeling
Sir.
of DNA. হ্যাঁ বলো।
স্যার এখানে স্যার হোমোজাইগাস আর হেটারোজাইগাস স্যার কোনটা কিভাবে চিনবো স্যার একটু বলছিলেন মনে হয় স্যার, আমি মিস করে গেছি।
হ্যাঁ, তুমি জাস্ট যদি তুমি রিকল করতে পারো, ফর এক্সাম্পল আমরা যদি এভাবে চিন্তা করি, get back to this picture, it will be easy for you. For example, if you have just take a single locus that is 15 and 20 repeats together. মানে একটা locus which should be heterozygous, and that heterozygous locus, the repeat number is 15 and 20. So for 15 and for 20, you are going to have two different amplified product. এটা আগে বুঝো। Is that okay? Is that understandable? যে 15 এর জন্য তুমি একটা লোকাস বা একটা ব্যান্ড পাবা, and for 15 you are going to get one band or one peak, and for 20 you are going to get another peak. Which one should be longer?
20.
20 should be longer. So just get a number of the size, a number of the length. Like 20, the band size should be like 300, and for 15, the band size should be like 250 or 270. So for that result, you are going to get in the gel electrophoresis 2 band. Okay. So for 20 size or 20 repeat containing product, it will, as I give you the number of the or length of the DNA, 300, and for the other one, the 15 repeat containing product should be like 270. So these 2 band will appear in the gel electrophoresis. Is that understandable? যে তুমি যদি heterozygous হয়, you are going to get 2 band.
জি স্যার, আলাদা আলাদা ব্যান্ড স্যার শো করতেছে স্যার।
Exactly. 2 separate size band indicate that this locus that you have checked is heterozygous. I'm giving you the number. One is containing 20 repeats, another one is 15 repeats. So obviously you are getting 2 different size amplified product, and if you run, you are going to get 2 different band. Is that okay? বুঝা গেল?
জি স্যার, বুঝতে পেরেছি স্যার।
ঠিক একইভাবে, when you are going to have this gel or this product run in the capillary electrophoresis, you are going to have 2 peaks. For example, one is this, take it as 280, another one should be like exactly on 350. Like this. Okay? বুঝা গেল? The 300 বরাবর একটা আর 280 just a bit like এটা 300 হলে বাদিকে. This is like from 0 to 100, 150, 300. So this should be 280 band or peak, and this should be 300. The same product, if you run in the capillary, you are going to see 2 peak for that specific PCR. Now, if you do understand this, so each and every locus that you are going to check, and you are going to get 2 peaks in case of a capillary electrophoresis for heterozygous result, and you will get single peak for homozygous result. Single peak কিভাবে হবে? ধরে নাও যে the same locus for another person contain 20 repeat in each and in both allele. তাহলে how you are going to have single size product. দুইটা allele, ধরো যে কোনো একটা subject, same locus contain 2 allele, and both allele contain 20 repeats, same number of repeats. What is the size of the band that you are going to get? In this case, you are going to get a single size band. That is 20 repeat, যেটা একটু আগে বলছে, it should be like 300 size. A single band তুমি পাও. You are going to get single band. Homozygous হলে single band কেন হবে এটা বোঝা গেল? Why you are going to get single band in homozygous?
দুইটাই হচ্ছে স্যার 20 রিপিট, এইজন্য স্যার।
Exactly. যেহেতু both the allele contain the same number of repeat, the product size should be same, and that's why you're going to get a single band in case of gel electrophoresis, and in case of capillary electrophoresis you are going to get a single pitch.
মানে স্যার এখানে স্যার এই 20 রিপিট যদি দুইটা থাকে তাহলে স্যার সেই ক্ষেত্রে তো ওভারল্যাপ করতেছে না স্যার একটার উপর আরেকটা?
ওভারল্যাপ বলতে কি আসলে তখন তুমি when you are going to do the PCR from both the allele, দুইটা allele থেকে it will do the amplification, and both the amplification will produce the same size product. So it should be like a single peak or single band. এটা দুইটা প্রোডাক্ট যেহেতু same size, as they are of same size, at the end when you run on the gel it will show a single band or single peak. Overlap, yeah, you can say it's a kind of overlap also, because they are same size, it will obviously produce same single band or same single peak.
Thank you,
sir. So for each and every locus result you expect, আগেও বলেছি আবারও বলি, you expect either 2 band or 2 peaks or single band or single peak. যখনই দুইটা peak দেখবা, whenever you see 2 peaks for any result, you can understand in this case means in DNA fingerprinting case, you can understand for that locus or for that PCR, that locus contain heterozygous number of repeat or different number of repeat. Or if you see a single peak, you can confirm that that locus contain same number of repeat or it is homozygous. Okay. Just for example, যদি আমরা এই ছবিগুলো একটু দেখি, যেমন ধরো এইটা, you can see this is a locus result, ML, just another locus name. You are seeing two peaks, দুইটা peaks দেখতে পাচ্ছ. What would be this ML result? Heterozygous or homozygous?
Sir, heterozygous.
Obviously true. Next one, D3S whatever. Next one is a blue color, যেটা এটা একটু বড় করে যদি দেখাই, ধরো এই যে এটা T3S1358. It also produced two peaks. What does it mean? It is heterozygous. In case of the next one, D8S1179, it produced two peaks again. So it is also heterozygous. D5S818, it is also producing two peaks. So it is heterozygous. This one VWA heterozygous. D21S11 heterozygous. Age D13S317, just a single peak. What does it mean? It is homozygous. ঠিক আছে, ঠিক একইভাবে FGA produce two peaks, so it is heterozygous. Acha, there is a confusion here. You can see this one D7S820, it is obviously a homozygous. D18S51, it is also heterozygous. এইখানে খেয়াল করো D13S317, here one peak and in between FGA it looks like another peak, black color, তাই না? So I think for this result D13S317 is also heterozygous, two peak. বুঝতে পারলা?
জি স্যার, দুইটা আলাদা আলাদা।
এটার সাথে মার্জ করে দেওয়াতে প্রথমে আমার খেয়াল হয় নাই, তোমাদের হয়তো বুঝে নাই। এটা it looks like two peak, one and another. এখানে black color একটা peak দেখাচ্ছে. So this result D13S317, this locus contain two product, so it is heterozygous. ঠিক একইভাবে নিচের ছবিটা যদি দেখো, heterozygous, this one heterozygous, এটা homozygous, heterozygous, this one homozygous. Very clear. বোঝা গেল সব?
Yes, sir.
Now question to you, why this peak is too long compared to this peak? It's not that long. What does it mean? So the yellow class বুঝে থাকো, you can understand, you can answer. প্রশ্নটা বুঝছো? যে this peak is long, this peak is short, this is also short. It is kind of like in between, medium size peak or medium long peak. It is also medium long. These are short long or short peak. Why is these differences? What does it indicate? একজন বলো।
Maybe size of the repeats.
No, size of the repeat is presented in this x-axis, this 100, 125, 150. So this has nothing to do with the size. Size is occurred in the x-axis. So the more farthest peak is the more bigger, or DNA size of that peak is more bigger compared to the more nearest peak to the zero. X-axis is 100, 120. These are the base pair size. Yeah, so this is 300 base pair, 275 base pair. So in this axis, this represents the size of the DNA. What represents the length of the DNA or height of the DNA? Height টা দেখি বুঝাচ্ছি। Y axis এ কি আছে? Last class যে বলছিলাম, in the Y axis you see 800, 1600, whatever the number. What does this increment mean? Y-axis এ বাড়তেছে. What does it indicating? This is the amount of DNA. Amount of DNA last class যে বলছিলাম মনে আছে কিনা? যেমন ধরো
Sir, concentration of DNA, sir.
exactly amount or concentration of DNA. এই যে this one. এই যে এখানে the same thing, the longer or the most long peak, medium size peak, and the shortest peak. This indicate less amount or less concentrated DNA, medium amount or medium concentrated DNA, highest concentrated. That's why it produce more intensity, and in this y-axis you are seeing the intensity. More DNA, more intensity. And that's why now you say, এখানে তাহলে what we are looking at, TH1 যেটা, is it present in highest concentration or lowest concentration?
Highest concentration.
Highest concentration. So TH1 হলো সবচেয়ে বেশি, মানে just the thing that you have to understand, the PCR for TH1 produce the highest amount of amplified product. The PCR of like this one, D7S820, produce the lowest amount of amplified product, but it doesn't like it do not make any change in the result. Any change in the result means whether the height is long or short of the peak, you can easily understand by looking at the number of the peak. These two is heterozygous, this one is also heterozygous, this one is homozygous, homozygous, heterozygous, homozygous, and homozygous. So height is not that important. You must have a clear understanding or have a clear result in terms of the number of peaks for each and every locus. If it is 2, you can say the locus is heterozygous. If it is 1, you can say the locus is homozygous. Now, just for example, just for your curiosity, I am giving you another example. Like for this position, TH01, you do the PCR of a sample and you get 3 peak. What does it mean? 3টা peak পেলাম, মানে 3 রকমের repeat আছে. 3 peaks means 3 different size of repeat carry in that sample. What does it mean? এটার মাধ্যমে তুমি কি বুঝবা? Whether the sample is good or sample is contaminated. যদি তুমি একটা PCR করো, if you do a PCR and you get like 3 peaks for that specific locus, what does it indicate? Is it a contamination of the DNA or it is okay? It is usual. 3 peaks for an individual result. Is it okay? What do you think?
Sir, I think it is a contaminated.
Exactly. কারণ আমাদের মধ্যে there is only 2 possibilities. I can get the same number of repeat from my mother and father, so I will be homozygous. I will produce a single peak for any locus or for any specific locus. I can get at highest 2 different repeats from my mother and father. দুইজন থেকে দুইটা ভ্যারিয়েশন পাইতে পারি, তিনটা তো পাইতে পারি না. So I can produce 2 repeats or 2 peaks in a specific locus. I cannot produce 3 peaks or 3 different repeat I cannot get from my mother and father. So it must be contamination. It is contamination ছাড়া অন্য কিছু না. That the sample may be contaminated and it carry 2 individuals blood DNA or blood together, or blood, I should not say blood, samples or DNA together. Okay. Any other question? If not, go to the DNA labeling process. Methods for labeling the DNA. The first method is very easy, is to use specific fluorescent dye, cyber green. We have already discussed about this type of dye in case of quantification. pico green method quantification এর কথা মনে আছে? Do you remember about the pico green quantification? The same type of dye is also used to label the DNA. When you do the PCR, use this type of dye. It could be cyber green, could be pico green, could be any other different color of dye. There are many different color of dye available. And here is a list of dyes. Many different dyes name is listed here. So I just want you to just read through this list and remember few of the name. That's all. It's nothing. Just there are so many different dyes are available now. You don't need to just remember. You just have to understand which different color of dye you have to use. You can get it from many different companies with many different name. So the main thing is this type of dye, cyber green, pico green, or this type of dye, or ethidium bromide, all this type of dye intercalate in between the double-stranded DNA, and it will label your DNA accordingly. So this dye, when remain free, it will not give you any fluorescent labeling or any fluorescent reading, but when it is intercalating with the DNA, it is going to fluoresce, it is going to glow, and you are going to see the color. And by looking at that color, or by this method, you can label your DNA. That is one protocol, the first protocol, and most cheapest protocol. Cheapest protocol, but as it is like not DNA specific. DNA specific means it do not differentiate among the different types of DNA. For example, in case of multiplex PCR, you are doing 4 PCR together and you are using cyber green. All the PCR product will be labeled with green cyber green color. So you cannot differentiate, at least with the color, that you are which one is first, which one is second, which one is third, and which one is fourth PCR result. You are doing 4 PCR result together in a reaction. You just add cyber green, all the amplified product of that multiplex PCR. It could be 4 PCR together, it could be 3 PCR together. All the products will be labeled with the same dye. So for that, as we discussed before during multiplex PCR, you have to have difference in the size of the product. Producter mode, you are doing 4 product together or 4 PCR together. All the PCR amplified product for this 4 PCR reaction should have different product size. So the same color will not be a problem. So alada size of it, you can get about the results interpretation very easily by having this different size product. Next one is fluorescence DNTP. You can use fluorescent label DNTP. Just for example, you can use DNTPs A, T, G, C. Among these, only a single nucleotide, take it as T, thiamine. Thiamine nucleotide could be labeled with your fluorescent dye. So all the newly synthesized DNA in the PCR reaction will be labeled by this fluorescent DNTP. So at the end, you are going to have very strong and very clear staining or labeling of your product. And all the different DNA, all the newly synthesized DNA that will be produced will be labeled with that fluorescent DNTP, and both the strand of the DNA. You see, both the strand. This is the primary forward strand, and this is the reverse strand. Both the strand will be labeled by the fluorescent dye. So this is good because it will freshly label your newly synthesized DNA. It is also good. The intensity or the labeling strength of this type of protocol is very strong, and it is also good because it will label both the strand of the DNA, and it is also good. It will only label the newly synthesized product. That's why only the amplified product will be labeled. In this case, or in cyber green case, whichever double-strand DNA is present, the template and newly synthesized DNA all will be labeled. But in this case, only the newly synthesized DNA will be labeled. So if you compare the first A and B together, B is much better. And as it is much better, you can think or you can assume that it is much costly, highly expensive of doing the protocol of B. It is costing you the highest among these three. Any question about this fluorescent DNTP labeling?
স্যার
হুম
স্যার এখানে স্যার প্রথম যেটা আর কি মানে প্রথমটা
হুম।
স্যার প্রথমটার ক্ষেত্রে কি স্যার মানে আমরা মানে যে সাইবার গ্রিডটা ইউজ করতেছি ইন্টারকালেক্ট করতেছে এই ক্ষেত্রে স্যার মানে প্রত্যেকটা কি ডিফারেন্ট সাইজ হওয়া লাগবে এই জন্য কারণ হচ্ছে হলো যে এটা যদি সেম সাইজ হয়ে যায় তাহলে ডিফারেন্সিয়েট করা যাবে না
Exactly. মনে করো you are doing 4 PCR together, that is multiplex PCR. And all the product, 4 PCR product, 4 different PCR, all has the same product size. Take it as 500 base pair. And you are using cybergreen. Cybergreen will intercalate among the DNA. It cannot differentiate PCR product 1, PCR product 2, PCR product 3, cannot. So it will intercalate with any DNA. So at the end, you will get all the amplification done and you will get a single peak. Just for example, 500 base pair, you cannot differentiate or you cannot say confirmly that PCR 1 is happening or PCR 2 is happening or PCR 3 or PCR 4. You don't know. All has the same size. You cannot differentiate. So for having or using this type of cybergreen, you must consider using the multiplex PCR according to the size differences. চারটা PCR করবা, চারটারই product size should be different with that. So that first product size like between 100 to 200. So in between 100 and 200, any peak must give you the first PCR result. For example, the second PCR result like 300 to 500. So in between 300 to 500, any peak will give you the confirmed result about PCR number 2. Third PCR, it may be like 600 to 700. So in between 600-700, you can get the third PCR result. So by this, you can differentiate among the PCR results, whether it is happening or not. হইলে পিক পাবা না হইলে পিক পাবা বুঝতে পারছেন
স্যার এখন যদি আমার স্যার ডিএনএ মানে সিকোয়েন্স গুলো যদি স্যার মানে সেম টাইপ হয় মানে সেম সাইজের হয় স্যার তাহলে সেক্ষেত্রে আমি এখানে কোন মেথডটা ইউজ করব
সেম সাইজে হয় যদি হয় তাহলে ভিন্ন কথা মানে it is difficult to then the last method is the best method. I will discuss this later, but you should consider all these things priorly. আগে থেকে চিন্তা করে আগে যাও. You have to consider everything. If it is same size, you can exclude those PCR which are same size. এইভাবে তুমি চিন্তা করতে পারো. Or if you don't have any other option, you have to use this PCR and it will produce same size, then you can think of the last one যেটা আমি এখন দেখাবো সেটা আসতেছে okay any other question if not so the last one is the most popular one that is using the fluorescent labeled primer now you can understand you are labeling the primer so just for example, you are doing 4 PCR in a multiplex reaction, and for each and every PCR, you have to use 2 different primers or 1 pair of primer for each and every PCR. So, 4 pair of primers. You can label each and every primer with different color. That's okay. Then if the product size is same, it will not make any problem. Even it is very rare that all the product size should be same. No, it is very rare. But if it is same, you can consider using this fluorescent labeled primer. For each and every locus, you are specifically using different primers, and those primers are just labeled with different color dye. For example, for 4 different PCR primer, you are using red, green, blue, and yellow. Then from the color differences, you can understand what is the result of your specific PCRs. In this case, what is good about this, you just have to order the fluorescent label primer and you don't have to worry about the product. But the disadvantage of this is you can see the first one and the second one. The whole product is getting labeled by the cyber green or the fluorescent DNTP. So the intensity of the labeling in first one and second one is very strong compared to the last one. Only the primer part, like you have just take an example like 500 base pair amplified product. Only the 30 base pair primer is labeled. But others newly synthesized product is nothing, is no labeling. So among the product, maybe you are getting only 20% nucleotides or 10% nucleotides get labeled. So the intensity of the labeling is much less, but specificity is very high. That's why it is very popular. বোঝা গেল?
স্যার এই তিনটার মধ্যে স্যার সবচেয়ে অ্যাডভান্টেজিয়াস কোনটা স্যার?
The last one. People are using the last one. The only disadvantage of using the last one, C, is it is not producing high intensity product, or the fluorescent intensity of this type of product is less. But as you are not looking at the product, how intense they are, the machine can even read a very faint reading. It is not a problem for the machine. So it is good. That's why people are using the last one, the fluorescent dye labeled primer, very frequently. And you see, it has very good, like the costing in the first and second, you have to use huge amount of fluorescent dye. The costing is much higher, the first and second. The last one, you just have to order the primer, like 30 base pair primer. Only those 30 base pair is labeled with the fluorescence. And others, all the products you don't need to label, so it will reduce the costing, and it will, as it is primer specific or PCR specific, you can increase the specificity of the result. That's why it is very important.
জি স্যার, এটা স্পেসিফিসিটি সবচেয়ে বেশি স্যার, বুঝতে পেরেছি।
Exactly. So you don't worry about the product size, you don't worry about any other difficulties. Just using this primer is very good enough, and it's also cheap.
স্যার আপনি যেটা বললেন স্যার, এইটাও তো একটা ভালো বিষয়। যদি আমাদের মানে আনফরচুনেটলি যদি ডিএনএ সাইজ সেমও হয়, প্রাইমারের মধ্যে আমরা ডিফারেন্ট ডাই ইউজ করলে ওটা ডিফারেন্সিয়েট করতে পারি।
Exactly. People are using this one from the beginning now. They don't care about the size also. But you know how to handle these things. Like if you don't have those specific primers, then you also have to think about how to go through with your like it's not research. It's kind of like diagnostic process. Everything is ready made. But as you're a researcher, you should know everything.
Yes, sir.
Okay, so these are the three methods that people are using. The last method is very popular, and not time. It should be like labor or not. It will be cost effective and more efficient in terms of specificity. That's why people are using the fluorescent label primer frequently compared to the other. Okay, let's end the detection protocol of the DNA. And this is the end of the capillary electrophoresis protocol. These are the picture. Like, okay, just get the difference. Here you can see just a single capillary. This is the like a first generation machine that people had. Now you also can buy a single capillary. What is the disadvantage of this machine is like, for each and every sample, you have to wait first sample to run first, then the second sample, then the third sample. So you cannot simultaneously run multiple samples together. So now you have like 98 samples. You need at least 1 minute for each, at least 100 minutes to finish. So it was a problem, as you have many samples nowadays, you have to do quickly. Then the companies now introducing multi-channel or multi-capillary electrophoresis. How many capillaries are here? You can count 1, 2, 3, 4, 5, 6, so 8 capillaries. So 8 capillary means 8 samples together it can run. So it is faster compared to the first one, the single capillary. Now we have 24 capillaries together machine, or the machine that can run 24 samples together. So by time we are in advancing our machines by increasing the number of capillaries. More capillary, more quick. More capillary, more quick. বোঝা গেল এটা যে the more capillary মানে বেশি capillary থাকলে you can run fast, তাড়াতাড়ি শেষ করতে হবে। যেমন ধরো this one 1 2 3 4 8 capillary machine. I think it's 16 capillary. দেখো খেয়াল করো, for each of this needle contain 2 capillary, 1 2 3 4 5 6 7 8 9 10, yeah, 16 capillary. So it is must be very quick, and we have 24 capillary machine. Okay, here we end the capillary electrophoresis part. Any question about this electrophoresis protocols? Oh. Okay, no question. Next topic is interpretation, data interpretation. It is interesting topic. Paternity testing and data interpretation in paternity testing. You know about DNA fingerprinting, one of the major function and one of the major use of DNA fingerprinting is paternity testing. I have already discussed with you. Now I'm asking you, if you have a baby abandoned in a garbage or somewhere, can you identify the parent of that baby? Question ta বোঝা গেল? So if you get a baby from a garbage or from somewhere, just a single baby, very young, just born, can you identify the parent of that baby by doing paternity testing? আলাপ মনে হয় করেছিলাম, বলো দেখি করা যাবে কিনা।
স্যার এখানে তো স্যার বায়োলজিক্যাল ফাদার তো মনে হয় স্যার মানে স্যার বায়োলজিক্যাল ফাদারের কথা কি বলতেছেন স্যার, নাকি মানে বাবা মা দুইজনের কথা?
I am saying about the parents. যে কোনো একটা, you are getting a baby out of nowhere, like no clue of its parents. Can you identify the parents of that baby by doing the DNA fingerprinting and matching the result within the database? Is it possible?
স্যার এটা তো পসিবল স্যার। এখন এসটিআর ওই বাচ্চার যে এসটিআরটা আমরা বের করে স্যার 13 লোকাস যদি আমরা স্যার পপুলেশন ডাটাবেজের সাথে আমরা যদি আমাদের যদি কোনো একটা ইয়ে থাকে মানে মানে আমাদের যদি কোনো সাসপেক্ট থাকে তাহলে সাসপেক্টের সাথে করতে পারি, আর যদি সাসপেক্ট না থাকে তাহলে আর র্যান্ডমলি যে আমাদের পপুলেশন ডাটাবেজ আছে ওইটার সাথে স্যার আমরা যদি মানে মানে ম্যাচিং
Yeah, I do understand your point. You are saying that if I have this baby and doing the DNA fingerprinting of this baby, I have the STR analysis done, and then I match it with the database and found a good match, kind of like perfect match. That person, that could be a male or female, is it legal to use that person as its parents? The answer is, it is not possible. It is not possible. Out of nowhere, you can identify parents of anyone. কোনোভাবে এটা সম্ভব না, and it is not acceptable. Even you get 100% match. 100% match পাওয়ার পরও it is not acceptable by the legal community, because to get a paternity testing done, you must have one biological parent available. একটা biological parent available, known confirmed biological parent must be available. Otherwise, even you get 100% match with someone, it is not acceptable in the court. Courte যাবে না, ঠিক আছে? So out of nowhere, you are getting a baby in a garbage and do all the process and get a match. It is not acceptable to the court or to the legal community. That this guy or that person, that could be a male, that could be a female, is the mother or father of that baby. It is not accepted. The thing that you have to consider, or you must have in your side, that the at least one biological parent must be present, and that is required to test then which one the other counterpart. For example, you have biological parent as father, then you can find the mother even from the database, no problem. If you have biological mother confirmed, then you can find the biological parent even from the database or from the suspect. That thing you must have to understand. You must have one biological parent, that could be mother or father, anyone, in your hand. Then you can go for the paternity testing. বোঝা গেল সবাই? Any question here? Okay, so you must have one biological parent, and as it is not finding the exact person, like in the crime scene or in the regular DNA fingerprinting application, you are looking for the same person. For example, in a crime scene, you get a blood sample. You are thinking that this blood may be from the criminal. Then you are looking for that criminal. You can check the blood samples result with a suspect or in the database. You get the perfect match. So in those cases, usually you are looking at the exact person. That's why it should be 100% match. In most of the criminal cases, you are looking for the exact person. That's why you must have 100% match. A single mismatch confirm that the two samples are not from the same source. We know everything about this. But in case of paternity testing, it is not that you are looking for the same person. You are comparing a baby or a person to find its father or mother. So to find its relative. So it is not mandatory, or it is not required that you should get the match 100%. In this type of paternity testing, you can take two mismatch as highest. Two mismatch means দুইটা mismatch to me you can take. Two mismatch as minimum two mismatch যদি ধরো দুইটা compare করলা and you get two mismatch, but all other 15, 16, whatever you checked, match. So you can say that, okay, this probably is the biological parent. But if you get more than three or more than two mismatch, like here it is written, minimum three mismatch or inconsistency have to be observed. তার মানে হলো to exclude or to confirm that that person is not the biological parent, you must get at least three mismatch. দুইটা পর্যন্ত, if you get two mismatch, you can still like confirm বলবো না, you can still make a probable result that that guy is the biological parent of this baby. But if you get three mismatch, you can confirm that that guy or that suspect is not the biological parent. Is that clear? The minimum দুইটা mismatch পাওয়া না, minimum তিনটা, you must have three mismatch to exclude any person from the analysis.
মানে স্যার তিনটা যদি মিসম্যাচ পাই তাইলেও আমরা বলতে পারবো যে এটা
না, if you get three mismatch, it is confirmed that the tested man, the tested man is excluded as the biological father. Barbar father লেখে এখানে, যাই হোক, biological father of the child. So if you get three mismatch, it is clear. You can 100% কি বলবো confidence say that the analysis is exclusion or the analysis is not matching to each other. So that person is not the biological parent. এটা তুমি বলতে পারবা. And as it is exclusion, you don't need, as previously discussed, you don't need any statistical analysis involved. No statistics is required there. But if you have inclusive result, like দুইটা mismatch পাইছো, বাকি you get just two mismatch, all other match, so it is inclusive. You get one mismatch, all other match, it is inclusive. You get all match, it is must be inclusive. So in those cases, you need to show a statistical calculation about the probability. As we discussed about the probability before, here also you have to discuss or do the statistical calculation. So এখানে যে জিনিসটা বুঝতে হবে, you are not looking for the same person, you are looking for the parent or you are looking for the children, whatever, paternity testing. You are looking for someone related. So you can absorb or tolerate some mismatches. And what is the mismatch or what is the tolerate level? At highest 2. দুইটা পর্যন্ত tolerate করতে পারবা. You can tolerate or you can take 2 mismatches. But if it is 3, you can confidently say that these 2 persons are not mother or these 2 persons are not biologically related. They are not son or daughter or they are not mother or father of each other. Okay? Any question here? If not, let's go to the interesting part. Here. This is the locus name that has already been checked. This is the data sheet. This is the mother, biologically confirmed mother. As I said, and as you know that one biological parent must be there. This is the biological parent. In this case, it is mother. This is the children. And this result is for an alleged person. It must be a male, which is alleged, which is a suspect as a father. Now you have to check. By definition, these children get for each and every locus 1 allele from the mother and 1 allele from the father. So these children should have 50% of its allele coming from that alleged father and 50% of its allele must come from this biological mother. Is that clear?
Yes.
Okay. If it is clear, now check one by one. First locus. The result of the mother is 1517. What does 1517 means? কেউ একজন বলো। 1517 for the first locus, result of the mother. What does it mean? কেউ একজন বলো। Is it heterozygous or homozygous?
Heterozygous.
Good. 1517 দিয়ে কি বুঝতেছি?
মানে স্যার 15 আর 17 ক্রোমোজোমে ব্যান্ড।
No. This is a locus that is D3S1358. Okay. In this locus, the mother genotype result is it contains 15 repeat and 17 repeat. ঠিক আছে? এই মাদারেরও তো ফাদার মাদার আছে। তার কাছ থেকে একটা মাদার থেকে আসছে ধরো 15 আর 17 আসছে অন্যজন কাছে। So together this mother is heterozygous. One allele contain 15 repeat and another allele contain 17 repeat. Okay? Clear? Now get the children genotype 1517 in this same locus. And the alleged person genotype is 1515. Now 15. Think about this one. 15. This can come from the mother or not? Bolo. I need interaction now.
জি স্যার, এই স্যার 15 তে তো স্যার বাবা মা দুইজন থেকেই আসতে পারে স্যার, কারণ দুইজনের তো 15 আসছে।
No, I'm asking only from the mother. As you are confirmed about the mother, I'm asking, or you should take the biological parent first. This 15 come from the mother. Is it okay? এটা কি কনফার্ম যে 15টা মাদার থেকে আসতে পারে?
জি স্যার,
15 may come from the mother. So think this way that the children's 15 is coming from the mother. Then this 17 must come from the father. It is now must come from the other guy. Other guy means the father. So this allele is called obligate allele. The allele that must come from the other person. In this case, the known person is biological mother. That is, just assume that is 15. So the 17 must come from the other person, and that's why this 17 allele is called obligate allele. Now look at the alleged father's result. Do 17 can come from the alleged father?
No sir,
আসতে পারে কি?
15 আছে স্যার।
So it is not possible. So what is the result? It is a mismatch result. Is it clear? 17 must come from the alleged father. If you think the 15 has come from the mother, 17 must from the father. But this father or this person do not have 17. So it is a mismatch result. If you take 15 coming from the mother. যদি 15 মাদার থেকে আসছে চিন্তা করো, 17 is not present in the father, so it is a mismatch. Is that clear?
Sir,
Bala.
sir, what if the 15 comes from the father?
I'm coming there. I'm coming there. First, you assume that 15 coming from the mother. Assume. Then it is a mismatch result. Understand or not?
Yes, sir.
Now it is a mismatch. Now it is also possible that 17 is coming from the mother. Now assume that in another way. Now assume that 17 is coming from the mother. Make it obscure. So 15 is now the obligate allele. It must come from the alleged person, and in this case 15 may come from this person. So now what is the result? Match or mismatch? If you take 17 is coming from the mother, what is the result? Match or mismatch?
Sir, match.
Match.
So if you take both way, you are getting one time mismatch and another time match. So it is a confusing result. So you should not take this allele result. Exclude this result. So this is not giving you a conclusive or confirmed information. That type of allele is usually termed as non-informative allele. Do you understand this? The A allele now, if you take one as coming from the mother, it is giving you a mismatch. If you take the other one as coming from the mother, it is giving you a match result. Match and mismatch together, so it is not giving you any concrete information. That's why this result or this allele interpretation is non-informative. And this type of allele just should be excluded from the study, and this is called non-informative allele. It is not giving you any conclusion. Once it's giving you a match, once it's giving you a mismatch. So it is non-conclusive. That's why it is non-informative. You just exclude this type of result. Understand everyone or not? Bala, প্রশ্নটা একটা করো।
Understand, sir?
হ্যাঁ, তাহলে এই টাইপের অ্যালিল যদি থাকে যে it will give you a match result once, it will give you a mismatch result and otherwise, so it is not informative, non-informative. Get it out of your
একটা
analysis.
কোশ্চেন আছে স্যার।
বলো,
এখানে আপনি যে বলছিলেন স্যার দুইটা পর্যন্ত মিসম্যাচ যদি হয় তখন স্যার আমরা অ্যাডমিট করতে পারবো।
সেটা আসবে কি? সেটা তো পুরাটা করার পর when you have checked all the locus,
আচ্ছা সব
then you get the result. It is now, what is the result of this? It is not even match, not even mismatch. Just exclude it. So you just exclude it. এটা কোনো কনক্লুসিভ রেজাল্ট যেটা দিচ্ছে না, জাস্ট এক্সক্লুড দিস রেজাল্ট ফ্রম ইউর অ্যানালাইসিস। এটাকে ম্যাচ মিসম্যাচ কিছু বলবা না। You cannot say it is match or mismatch. So just exclude it. Now move to the next locus. For example, this one. Look at this. 16, 18, 16, 18, 16, 18. All 16, 18. Now again think that the 16 in children is coming from the mother, biological parent. If you think 16 is coming from the biological mother, 18 must come from the alleged person. So it is a match. In other way, if you think that 18 is coming from the biological mother, it is possible. Then 16 must come from the alleged father, and it is also a match. So in both direction, you are getting a match result. So you can take this result, and this type of allele is called informative allele. এটা খুব সহজে বোঝা যাচ্ছে যে in either way you are getting a same result. Same result means match result. So you can take this result, and in this case it is match, and this type of allele is called informative allele. Any question? এটা কি বোঝা গেল? Okay, I think you understand it. Now move to the next one. These two are most difficult one. These are very easy. Now you can see these are easy things to get. For example, in this locus, the children genotype is 11-13 and mother genotype is 9-11. So 11 must come from the mother. So here it is no confusion that 11 is coming from the mother. The 13 must come from the father. And you see in the father it has 13. So it is also a match. It's very easy to understand. Now move to the next one. In case of children, you have 24, which must come from the mother. This 24 of mother is giving to the children. Then this 17 is the obligate allele that must come from the other guy or other person, and 17 is present in the father. So it is also a match. Very easy to understand. Next one.
বাট স্যার এইটার যদি আমরা উল্টায় দেই স্যার তখন তো আবার মিসম্যাচ হচ্ছে।
উল্টায় দিবা কিভাবে বাবা? উল্টায় দিবা বলতে
আমরা যদি স্যার এই যে আপনি এইটা বলেন যে 24 যদি মাদার থেকে আসে আর 17 যদি ফাদার থেকে আসে তাহলে স্যার
24 is not if, 24 is must.
জি স্যার, এখন যদি স্যার আমি বলি যে স্যার এখানে 17 হচ্ছিল মাদার থেকে আসতেছে আর 24
কেমনে বলবা? Do the 17 present in the mother's genotype?
না স্যার, নো স্যার।
তাহলে তো you cannot say that. That's why you need a confirmed biological parent. এখানে mother. বুঝতে পারছো?
আচ্ছা আচ্ছা।
That's why you need a confirmed biological parent. This 24 must come from this mother, because 17 is not present in the mother. The source of 17 obviously is not the mother. 24 must come from the mother. Okay, now this 17 must come from the other guy, and in this case the other guy also carry the 17. So it is a match result. Parishkar?
জি স্যার।
Next one, check. Again, the 14 must come from the mother. 14 must come from the mother. Calculate. ঠিক আছে? Because 10 is not in the mother present. 14 must come from the mother, and 10 must come. 10 is the obligate allele. It must come from the other guy, which is the alleged person here, father, and it also carry 10. So it is also a match result. Move forward to the next one. Yeah, 31, 31, 31. So this 31 must come from the mother, because mother has only 131. So at least 131 of these children is homozygous in this case. At least 131 must come from the mother, because the mother has other genotype, that is 30, and it is not present in the children. So this 31 must go to the children. So just assume this one is coming from the mother for sure. No other chances. So the other 31 is the obligate allele here, and it must come from the other guy. Look at this guy, it carry 31. So it is also a match result. Any confusion here?
স্যার এগুলা কি স্যার যে ম্যাচ হচ্ছে স্যার এগুলা কি ইনফরমেটিভ নাকি নন ইনফরমেটিভ স্যার?
All those giving you a confirmed result are informative, whether match or mismatch.
তাহলে স্যার প্রথমটা
ইনফরমেশন দিচ্ছে।
ইনফরমেটিভ হওয়ার কথা স্যার, একদম ফার্স্ট একটা।
এক্স্যাক্টলি, হ্যাঁ, প্রথমটা কেন ইনফরমেটিভ হবে? ইট ইজ গিভিং এ কনফিউজিং রেজাল্ট। একবার 15, যদি ইফ ইউ টেক মাদার, if you take 15 is coming from the mother, it is giving a mismatch. If you take 17 is coming from the mother, it is giving a match. So one time it's giving you a match, one time it's giving you a mismatch. Confusion. Which one you want to take? You cannot take both. So it is non-informative.
ওকে স্যার।
বুঝতে পারছো?
Yes.
প্রথমটাতে তুমি ম্যাচ মিসম্যাচ দুই রকম রেজাল্টই পাচ্ছ। So you cannot take both. So it is not informative. Non-informative, that's why. আর এইটা যেটা এখন দেখাচ্ছি 30, 31, the thing that you are looking at. Any question here? One of the children 31 must come from the mother. The other one should come from the other person, alleged person. Alleged person also has 31. So it is a match result. Any confusion here?
No.
নো স্যার।
Okay. Now you can see one by one, all will show you a match result. Like 17 here coming from the mother, 13 should come from the father. It is present in the father. Next one is 14 should be come from the mother, because only 14 could come from the mother. So 13 must come from the father. It is there. Then the next one, 21 must come from the mother, and the other 21 should be from the father. The father also carry 21, so it is a match. And the last one, 22 must come from the mother, 24 should come from the father, and it is also showing that it is present in the father, so it is also a match. So by comparing all these, how many? Let's say 10 data, only one is non-informative, the first one. But all others are informative and all give you completely match result. So at the end, you are not getting any mismatch, all match. So that's why it is an inclusive result, and it is concluding that this alleged father or this alleged person is surely or almost 99.99% confirmatively sure that this is the guy who is the biological father of this child. Okay. Okay, let's play with others in at home. We will start from here from next class. Stop the recording, Daniel.
Okay, sir.